Good afternoon, everyone. Thank you so much for coming and joining us this afternoon. On behalf of everyone at the Center for Brain Health, welcome to Frontiers of Brain Health, where we take a deep dive into the most exciting new developments of brain research. I'm Francesca Philby. I am a professor of cognition and neuroscience and the director of the Laboratory of Neuroimaging of Reward Dynamics here at Brain Health. I'm also Senior Associate Provost at UT Dallas, where we are starting a postdoc mentoring program. I want to make sure that everybody knows that we do have a postdoc mentoring program at UTD, so please make sure to reach out. For today, I just want to remind everyone that we will have a few minutes at the end to answer some questions. So for the virtual audience, please use the Q&A function, and for those here with us in person, have your questions ready to ask at the end of the talk. We'll get to as many as we can at the end, and this presentation is being recorded. For those who don't know much about us, the Center for Brain Health is a cognitive neuroscience research center at the University of Texas at Dallas. We have dedicated the past 3 decades to exploring neuroplasticity and the brain's amazing lifelong potential to get stronger and work better. Now I would like to introduce you to a wonderful speaker, Dr. Noah Ofen. Dr. Noah Ofen is a professor at the Center for Vital Longevity at the University of Texas at Dallas in the School of Behavioral and Brain Sciences within the Department of Psychology. Dr. Ofen joined UT Dallas last year? This past January. So she's relatively new, so please make sure to welcome her. Having held a faculty position at Wayne State University in Detroit, Michigan, her research interests include developmental cognitive neuroscience across the lifespan, human memory, spatiotemporal dynamics of brain function, intracranial EEG, functional MRI, high-resolution imaging of the human hippocampus, genetic and environmental influences on cognitive and brain development. So I look forward to your presentation. Welcome, Dr. Ofen.
So it's wonderful to see so many of you here. It's really a fantastic venue to speaking, right? And it's really inspiring to kind of get the brain going and all this spiking activity that's kind of represented right above us. So let's hope I can keep up to that feel. Everybody hears me well? Perfect, wonderful. So I always like to start my talks by showing pictures of my kids. Really in order to say, a child comes into the world with very limited ability to interact with it. And over the process of unfolding of many developmental processes, they learn to walk and talk, run and climb, and through more formal education, they learn to read, write, do math, play chess, maybe play music, and as I like to say, get to the apex of existence, and this is engaging lifespan cognitive neuroscience. Again, as I said, a lot of developmental processes are unfolding during this. The main research question that I'm trying to get through, what is happening and how can a child acquire such a vast array of abilities, is by asking how does this vast array of abilities, the cognitive development, is supported by the development of the brain, which undergoes robust development in every measure that we might want to look at. So, in order to understand cognitive development, my favorite domain, and almost like my kind of animal study or animal model, is memory. And I love studying memory because it allows me to do so many different things. In order to remember information, you have to process information, maybe visually, maybe auditorily. You have to then somehow store the information and be able to, upon request, follow some task requirements and be able to have enough of executive functioning to actually provide the correct answer. So I study memory, but it really allows me to look at the very vast phenomena within cognition. But study memory development, so one of the first things we'll do, and this is in my talk at the beginning, would be asking what is memory development? What develops in different developmental timetables. But then ultimately, I'm going to ask, and I'll put this question later on, is what is this memory development, the development of, with the proposed answer, the brain, then analyze it, and I'm going to use several different measures of domains, so ways to kind of quantify brain, starting with looking at brain structure, which I would say provides the underlying limits, endogenous limits, or that enable or limit cognition. And then I'm going to switch at the last part of the talk to talk about two different ways by which we quantify brain function. In both cases, our goal would be to link brain structure to cognitive development, brain function to cognitive development. But before we start, so we can kind of start thinking about memory and memory development, let me challenge your inner child to a memory game. And this is from, again, my kids, from outreach events in which we practice. And then they start to be the ones who administer like these fun little games for people in the data. So I'm gonna give you a couple of memory games. Ready? Try to study this as best you can. It's gone. I'm gonna flash another display. Tell me what differs in the new display. Exactly. So if I put them one right next to the other, the belt is now an aquarium or fish tank. So that was fairly easy. Next game. Study. Gone. What has now changed? Now we have the hand instead of the pendulum, but something else? The location of the basket and the beetle has now also switched. And that was already, you can see, based on like not many of, I made it a bit harder, I'm kind of flashing very quickly, but you see that there's already two different things that you might have been remembering, two different qualitative aspects of memory. One of them might be harder than the other. And then another dimension by which study this one, I'm going to give you a little bit more time. Ready? It's gone. And in this one, what has changed? What has not changed? Something was so if any of you said something hasn't changed, then they would be wrong, because indeed the right answer is everything. And by everything, what I've done is I replaced two items, so that's the item memory that was maybe easier to pick on. I did like a one switch, but also I switched the location of all the other four items in this state, right? So as you can see, memory can be easy or hard, depending on what aspects of memory you're tapping into when you measure what memory might be. So in the examples I gave you here, we've had memory for single items, memory for associations, and then you can manipulate the number of details. And all those are examples by which you can start thinking of memory not as a unitary entity. There's many different ways. And of course, this is just scratching the surface of like how we think about memory, many other phenomena. These are all memory that we can qualify as recognition, and can you recognize what you've seen before and match it with the correct information that you need to judge whether it's the same or different. But still, those single items versus associations and number of details, I dwell on them. I would need to, I suspect that this is not the correct one. So I'm going to quickly switch over. Sorry. Yeah. And I'll get back to the correct one when we get there. And it's working. Okay, perfect. So I dwell on this because when we are trying to quantify memory development or to assess what some aspect that may show more protracted development, the development of those unfold over longer time periods versus other aspects that are more mature earlier on, those distinctions become very relevant. So if you look at within the age range that most of my work has been focused on, and this is from middle childhood through young adulthood, you'd find that memory for items in these cartoon examples, and soon enough after this I'll show you real data, is fairly stable. that is within this kind of middle childhood, you get pretty good at the simple way of doing memory, whereas those types of memory that I would ask you for the exact location of the association between items, and I increase the number of details, this is where we see that children underperform, and those adults kind of perform better than children. So now we have ways to quantify memory, and this is research that we conducted at many labs. This is how I synthesize it. There's some aspects that are more impoverished, more simple, easier, and we find those memory aspects that are stable, and others would show more protracted development. And one way to show it with data that we actually collected in the lab over the years is by making a paired associative word pair associative task. And participant would study pairs of words, and then we tested after a short, brief instructor delay on either single words or the exact pairing. So it would be words that I've seen before that were either paired with each other or paired with other words before. What you can see here again, just as a way to show you the schematic comes to life with actual data, is that remembering the single words would be fairly stable within this age range, whereas remembering the exact pairing of the word pairs is what children would show lower performance compared to the adults. So this is just one example in which we have items versus associations showing differential developmental trajectories. And just one, so we kind of took a much wider array of tasks in which all participants took that task and many others, and then we put them together in order to better quantify this item versus associative component and get a better handle of how to quantify developmental differences. And this is what you see now when we use the structured question modeling and a lot of indicators that load onto those now latent constructs and give us a much better assessment of item versus associative memory. And indeed, for the single items, we find that now it doesn't stay flat. Now we have within our younger participants, we still have some development, but then it plateaus. So we do have some of the stability in this non-linear trend, whereas for looking for the specific pairing of the words, we still see linear increases, indicating more protracted development. So this is some of the more recent work, but overall, we characterize memory development as you say there are some aspects that are more stable, others that show protracted development. And now we can ask that question that I've already alluded to. I'm going to give you at the very beginning, which is what is memory development, the development of? And I always like to say it's not just that English is my second language, that I talk weirdly. This is actually the title of a very influential symposium in Society for Research in Child Development in 1971. That's what they asked back then. What were the answers? You can kind of start thinking for yourself, and then for the interest of time, I'm going to give it to you. But I'm first going to say, for sure it wasn't the brain, right? Because then we didn't have access to much of like trying to understand the hardware development that support this cognitive development. But they had really good ideas that we're still following. For example, they say that perhaps the memory develops because a child has more knowledge about the world. So the knowledge base would allow you to kind of string things together and put them in better structures, and now you have better. You also might have better strategies to put those things together. So these strategies, strategic control strategies would be another thing. And it just may have better ability to keep more information in mind, the capacity of working memory. All those kind of things were like the answers. This was kind of the three talks I assume. As far as I understand that symposium, and I have met people that participated in this. It was really fun discussion. It was in a talk like this, and so I'm like, I was there, and so we had lovely discussions ever since. So we're still interested in this, but as a cognitive neuroscientist, what I'm trying to suggest is that memory development might be underlying, as I told you before, by the development of the brain. And we're going to use those three different measures to get us thinking about different aspects of brain development, which aren't really distinct, right? But we have those measures as distinct measures, so we kind of pull them out and look for the correlations as best we can. So starting with brain structure. And by the way, still my kid in this picture. So as a pilot subject in one of my studies. So starting with brain structure, the brain changes. Whatever we can quantify for measures of structure and differs one by age. Changes is what we can say if we actually follow the same brain. But overall, typically people just look at age differences. So there are age differences in any structural measures. And this is a really nice paper that came out now I guess 2 years ago, that took 100,000, if I remember correctly, data points for many, as you can see, samples. I think I have it. It's not an order. It is that many to try to quantify some differences across the brain over years with many databases. This is mainly cross-sectional, but it has some longitudinal. It has a lot of beautiful things. But what you can see is that across different measures of brain structure, there is a lot that is happening throughout the life. But I can just do this. But I'm going to clue you into where I work and what I'm interested in. This is from middle childhood-ish throughout adulthood. You see that there's robust changes, robust differences that happen during that age range. And with respect to the gray matter, the cortical thickness, or the gray matter volume, as well as white matter volume, that seems to kind of still do a lot of change, undergo a lot of change. And this is in a global, across the whole brain. If you look at other earlier work, but still foundational and still very much the case, accepted, you see that those age differences that you find are not, this is from a small longitudinal sample, so you can talk about change. You can see that the bluer it is, the thinner the cortex, this would be the blue line here, but you see that it doesn't happen at the same rate throughout the brain, right? So now we know that the brain changes, we know that it doesn't happen at the same rate, and we want to ask how does changes in the brain might underlie changes in cognition. So what we do is we look into the brain, and specifically we care about memory. I care about memory. So I started by putting the most key regions that are kind of key suspects for what do we care about, where would we look for how the brain supports memory. Development is by looking in where the brain supports memory. And the brain supports memory. Critical for memory is the medial temporal lobe, including the hippocampus, lesion of the hippocampus, resulting inability to remember new facts and events. So global amnesia due to lesions in the hippocampus, critical for memory. But lesion studies, throughout and summarized in the 80s and before, have also identified that the prefrontal cortex is important for memory because lesions in the prefrontal cortex lead to the type of memory deficits that are very similar to the more complex associations, more details. Remember the order by which you represented the information. Who did you hear it from? A female voice or a male voice? All those are lesions. Individuals with lesions in the prefrontal cortex show specific deficits. Okay? So we have two regions we care about, and starting with the prefrontal cortex, let's see, and more so interestingly to say, I showed you it's not in the same rate, and you can see that perhaps it gets bluish, bluest, later on when we talk about the prefrontal cortex. So it's not just that it has supporting those type of memory that is most protractedly developing, it also shows the most protracted structure development. So we're trying to put this together by picking something that the prefrontal cortex we're told are helping us do, and this is keeping in mind a list of words. Say, like you go for shopping and you want to say, I need to get this and this and this. Keeping this information in mind and being able to reflect on this information is something that would be supported, among other things, by being able to use organization principles and kind of the strategies that the prefrontal cortex might support you doing. So we asked, are age differences in prefrontal cortex volume linked to something that relates to memory? In this case, is the use of strategies, keeping in mind some good mnemonic strategies to remember more information. And the way we've done it is by using the California Verbal Learning Task, CVLT. This is not the actual words in the task, but just as an example of how the test would be administered. Participants were read a list of words and then had to free recall as best as they could, as many as they could. They do it over several times so it can have opportunities to kind of boost their performance. But what's key in this specific task is that the 15 words, it was the child version, came from 3 distinct object categories. Here you can find the same kind of thing. So when you recall, if you string together, if you chunk together words that came from the same category, that is a strategy that is useful for you to remember more information. And so in this example, you see that if there, in this recall, we can quantify, we could have, if that was an example, we could quantify that there are more chunks of what we call semantic clustering. So you cluster together words that came originally from the same category, and that might help you remember better. So that specific strategy, indeed, adults did more so than kids in that study. So children did use less semantic clustering, and the more the individuals used semantic clustering, right, the thinner or the less volume of specifically these prefrontal cortex regions they had. Remember that we have for this region thinning. So now we have linked a physical brain measure. This is like the volume of prefrontal cortex to ability, is linked across individuals to ability to use mnemonic clustering, in this case semantic clustering. And we were able to show that it's partially accounting for the age-related increase in semantic clustering, and this volume of that specific region in the prefrontal cortex was related to. Okay, so partially, but now we find one way prefrontal cortex we think is going to evolve. Yes, some structural feature in the prefrontal cortex supports age-related differences in memory, in this use of semantic strategy. So we can say yes, prefrontal cortex. Now, like we say, age difference in prefrontal cortex volume linked to age differences in mnemonic. Differences in mnemonic start to use. But I said, what's really crucial to memory is the medial temporal lobe, including the hippocampus. So let's think for a second about the development, the structural development of the hippocampus. Again, from like early and foundational work, identify that the hippocampus volume itself doesn't show much differences. So the total volume of the hippocampus is fairly stable across age. But what this kind of first foundational study also I highlighted is that there might be some more subtle differences in sub-regions, or as we say now, as we know more about the development of the hippocampus, the subfields of the hippocampus. The hippocampus is a complex structure, and the subfields of the hippocampus are crucial functional organizational distinction. So we are going to ask, are age differences in hippocampal subfield volumes linked to age differences in memory? And to do that, we spend a lot of time, and we still are spending a lot of time optimizing. By optimizing, I mean harmonizing across labs and making sure that we are staying as close to valid measures as we can and keep a really high level of reliability in our measurements. So we have actually a way to quantify hippocampal subfield volumes. So we were able to do that. And then we went back and said, within the hippocampal subfield volume, I said it's functionally relevant. And it's actually some parts of the hippocampus are thought to be more related to this associative memory versus item. So we took the same, you've already seen this, and now we have quantification of individual differences in associative memory and item memory. And we took our careful volume estimates of the different hippocampal subfields that we could quantify and put this all together to tell us where the variability kind of plays together and kind of hangs together. And what we were identifying, if we look for the age, as I already told you, age increases for the associative memory, not the item memory. And indeed, it was the age differences in the volume of that specific hippocampal subfield that we know is related to associative memory. We've done a bunch of many different theories exactly how it is, but we know it is more expected to be related to associative item memory. And indeed, age, there was a specific effect that showed that age differences in the volume of that region was explaining, there was a direct path explaining age differences in associative memory, but not item. So great, we had another answer for the second question. Yes, we can find specific effects. It's not the end, because as I said, the just quantifying hippocampal subfields is pretty challenging and take the efforts of a large group of individuals from all over the world that come together. And we've been meeting since 2013 pretty regularly to get to the level that we can have a harmonized protocol that everybody agrees on, that this is how we should be segmenting the hippocampal subfields. And we made that much progress that the next paper that's coming up would present a reliable protocol for the body section of the hippocampus. The head is still too complicated. The tail, forget about it. So it's a lot of effort, but because this is really important, if we want to do this, to do it in a way that is accepted across individuals. Within our lab, we contributed to validating, showing that these type of protocols are reliable, and we've done this by doing a test-retest reliability. And then, because we know it's reliable, we say it would be a great way to quantify any changes. This is over 1 month, this is over 2 years, and you can see that there's not much that's changing because it's a fairly stable structure, but there is variability in the change. And because those are reliable measures, we can take this variability and try to link how it relates to variability in cognition. And I said over 2 years, so just to say, structural measures are really fantastic for looking at change. I said it's stable, and this is how much stable it is. I'm going to leave it on for a second for everybody to get the impression. This is the same individuals coming back 2 years later. You can see it's the same person. Right? It takes some time to the untrained eye, but you can see that the shapes are very similar and different across individuals, but it's the same person, right? So that's really nice. This is something that's harder to get in more functional measures that are influenced by so many properties. But it's really important to have good, reliable measures to quantify those differences in that change. And longitudinal studies are really critical, but rare. What's also rare and would be of great interest for us to those who are not just interested in development is to try to understand how those changes, differences are panning out across the whole lifespan. And for that, in my lab, we did some meta-analysis to show that there are across many different studies, not that many, it's still coming up, developmental studies, there's some effects of showing maybe some different hippocampal subfields are indeed still undergo development, but the effect is very small. In aging, it's much more robust and much more consistent. Everything shrinks. Right? So trying to put together how is those critical to memory regions in the hippocampal subfields related to the development of cognition also has very important thinking about at the other end of the lifespan. But these are cross-sectional studies, everything that we reviewed. And indeed, just from these cross-sectional studies, you can think, as I've shown you before, their memory improves. This is a study we published a few years ago across the whole lifespan. These are very, very rare types of studies. But this is, again, cross-sectional, so we can see that the volume differ, performance differ. We assume that there might be some relation, but to get to that relation, what we really have to have is those lifespan longitudinal studies. So the more we think about it, the more we collaborate among us and think together, the better it is. And what you see here is current findings that we have showing behavior on the right hand and brain volumes behavior on the right, on the left, and brain volume for this specific hippocampal subfield that we care about on the right. And now we'll be able to go and ask, okay, the change here, how does it link to the change there? We will be able to find much more unique effects, and this is kind of this certain associations, and we can look them together. This is just a nice comparison with what we can kind of just see and have no idea, and maybe talk a lot about how it might relate, but we really don't know. Now we'll be able to do this. What else this hippocampal subfield volume, and with the generosity of other labs that have done the same type of things, is take our questions about the sources of individual differences to the next level. One evidence we have from my lab is that socioeconomic status does seem to relate to hippocampal volume, and specifically, we found in the sample we had with kids from age 8 to 25, or maybe even 5 to 25, that there was some relationship with socioeconomic status. An odd one here, because we find that higher hippocampal volume was related to the lower socioeconomic status. In a developmental context, we can come up with a lot of ways of why that might be. If we are expecting pruning, if we are expecting maturation to actually look at smaller volume, maybe there will be some differences there. But this is a finding that we've had to kind of get better at addressing the source of individual differences in brain volume and how those might relate to cognition. We are partnered with a number of different labs, and this is work from Kelsey Canada, postdoc in the lab, that each of the lab uses different methods. So how do we even bring all this data together? We can reprocess and just have one sample, but even better is if we take advantage of all the good but different methods across the different lab. And this is hot off the press, so I had to kind of put it here for just a second. I got the notification to share my new paper today, and this is an integrative data analysis approach, which is taking from like other schools of thought that deal with a lot of data and try to kind of integrate things that aren't exactly the same, but they're close enough, they're all kind of valid in their own way, and we want to get the general insights from this. So the integrative data analysis approach, what you can see here, kind of matches, but the colors are different, so I moved it to here. This is from a data set that we collected in Detroit, and then we combined it with data collected in Davis, and with data collected in Maryland, College Park, which had the same hippocampal subfields, and we did all integrated data analysis, making sure that we can put those together, even though they're slightly different protocols, different ways by which to define what the different subregions, still valid and good, and with this we can find effects that are consistent with what I've shown you from the meta-analysis, for example. So we have age differences that are specific to one of the subfields that we've done. So this is just because it's off the press and shows to the extent that excitement of that we can do relying on structure. And I've mentioned structure will be the endogenous constraints on memory development, and shown you several examples. And then I would just highlight that, of course, there are many more questions. We don't really know what the volume means, right? But the volume is such a macro level measure that we assume it relates to these neurobiological processes that lead to it, but we can't very well map it. There's so many things that are happening that lead to differences in volume. And it's not a really direct measure of what the brain was doing that relates to memory. Comes brain function, other toolkits, other tools in our toolkit that allows us to get this more direct measure. What's really nice about functional neuroimaging is that it does give us, we can see the brain all the time or whenever we look at it with these functional measures. So in the case of memory has different phases. You encode information, then you store it, and then you retrieve. With functional neuroimaging, you can actually try to take a snapshot of the brain during encoding, maybe even try to understand what this storage means in the brain, and for sure you can look at the brain as it tries to retrieve information that you studied in the past. So this is beyond just looking at performance and getting this gross measure of performance linked to some variability we have with structure, which would be the only way to go that way. So my favorite way is not through the memories games that we played in the beginning, but another kind of memory game, the interest of time, let's go through. I'm going to show, we're showing participants pictures of indoor and outdoor scenes, and all they need to do while they study them, two things they need to do: tell us whether it's indoor or outdoor, and try the best they can to remember them, because they know we're going to test them for all the scenes they've just studied. Why do we also ask them to say indoor outdoor? Because we want to know that they paid sufficient attention to the task, so that if there are scenes that we didn't get any response, or the wrong response. We can get these specific time points out of the data analysis, and then we can say children, adults, whoever we studied paid sufficient attention to get this encoding task correct. So we do this, and then we take them out and ask them to remember it. Some they do, some they don't. This would be an example or summary of all the many participants we tested. Open circles, fMRI studies, blue dots, these are intracranial EEG studies I'm going to talk about later. That task is a bit easier. That's why the performance is kind of still win and maybe even above. The line is referring to the intracranial patients, but as you can see, there's a lot of variability there. And that's I always pause earlier in the talk and say, development, sure, we can find it, but look how big the cloud around the line is. So individual difference is really our bread and butter. We have to be happy with it and use it with the methods that I've shown before. So performance shows a developmental difference, but then with intracranial, with the functional MRI, we can go back into the time points where the brain was seeing a scene that later remember versus forgotten, and make this contrast between what was subsequently hit and subsequently forgotten as our memory contrast. So we can actually see what the brain is doing for memory. And again, we're going to be focusing on those two regions, and with functional imaging methods, we can ask the main question people have been asking is where. In this case, where in the brain are age differences that support memory development? Right? But really what we want to know is how. How does the brain does it? Right? So we start by, we know what the prefrontal cortex might do, the medial temporal might do. So we answer where, and we think we have a better handle on the how, but the how is really always what we do. So starting with the where, that's the very first study I published. You can see that when we look at this subsequent hit versus subsequent miss, what did you do that you later remember compared to later forgotten when you studied an item? And you can see the regions, the medial temporal regions over here, and regions in the prefrontal cortex. And we compared across individuals, we got a very clear pattern that was very exciting to us and consistent with the idea that the medial temporal might be more stable. And at the time, we weren't very careful. It's still very difficult with functional MRI to get to the resolution of subfields within the hippocampus. But there were activations in all age groups or across age. What we did see that increases with age and support memory for richer information is the activations in the prefrontal cortex. So for that very first study, we said, where are the age differences that support memory development? Prefrontal cortex. Remember, that's also the region that shows more protracted development. So that's a great answer. And we stuck with this and followed up on that for a lot of time. But this is really just the where. If you want to go a little bit beyond the where, still within very similar methods, we simply open our eyes to say, this is what was more active when you later remember compared to forgotten in the previous slide. This is still here in the yellow-orange. But there are also regions that are less, as in what they do is they more deactivate when you later remember compared to you later forgotten. And those are the blue regions, and those also show really robust developmental, even more bigger age differences in how much of deactivation you get for what you later remember. What might that be based on its location and like how it connects to the rest of the brain? We say like maybe what those regions do that providing more effective something like a thought suppression that allows the child to remember better information, allows the adult to remember better information later on. Another way to go where is to start to ask how those regions connect with the rest of the brain and not just have a region but a more towards a network type of analysis. And indeed we found that from both of those regions, they are connecting in a way that supports memory development. And I'll stick with this at this time. So this would be the way by which both of these thought suppression regions, they interact with the medial temporal lobe more so with age. And the positive subsequent memory, positive effect also connect more with the medial temporal lobe with age. But so this is kind of tricky ways in which you can start to get into the how. But I was always not fully happy with this. So enters a really, really exciting, rare opportunity to take advantage of patients which they have electrodes implanted in the brain or on top of the brain for the clinical management of epilepsy. But during the process, they're in the hospital. They are able to participate if they would be asked to and willing to in very similar, in experimental tasks. And very simple task is even better. And the task that I loved for so many years is that you don't need to do too much. Just see a scene and you try to remember this. You remember, maybe you forget. So what we've done is we use this intracranial finding. What you see here is an aggregate of over 50, I think at this point it was 67 data cases, data sets that we've collected over many years. So I think it's more than 10, about 10 years of painstakingly finding those patient population and being able to aggregate. But then each of the individual patients would have just some specific coverage, but across all of them, all those little blue dots would be dots that we can get data for, and the data would look something like this, and we can do so much with that data. So what does intracranial EEG gives us is to get into what are the underlying mechanisms that coordinate brain activity within a region, between regions, and ultimately, because they're doing memory tasks, we can ask how are those supporting memory development. Because this is a memory task, and we have participants across a wide age range, we can ask how does this support memory development. And this is really exciting because, like fMRI, we can kind of ultimately across everybody get great localization. And like EEG, we have access to the information, it's electrical activity. That's much more direct measure of the neurons or the region's activity compared to fMRI. So this is how an example participant might look like. This is where we had with my collaborator at Wayne State University, where they used to do a lot of greens and strips. So these are electrodes that are placed on top of the brain. And it will be on one side of the brain, but because the task that I use is perfectly fine, you get activations, whether it's left or right, it's not very lateralized activations we have. That's what we find in fMRI as well, so we know it's going to work. We're going to get this. So we have a great location, and then we have this temporally and spatially precise recordings of memory-related activity. And we can ask things about timing of localized neuronal activity, which we can't test with functional MRI. We can look at both the local and regional coordination and the different types of mechanisms that coordinate activity across regions. Different types, there's multiple things we can do. We can look at the high frequency activity. We can look at the oscillatory frequencies, different types of oscillatory frequency. They have a phase and they have an amplitude. So we can look at all those different things together to try to isolate what is it that support memory in the brain, and what of those differ by age. And for example, we can even ask very basic questions, because we know brain development, but just starting to get more idea of how does those oscillatory rhythms develop in the brain. There's knowledge that has been ongoing for a long time, but hasn't been quite directly related to the core cognitive neuroscience. Okay, so we look at, for example, how in this first study we published, we looked at the prefrontal cortex, or you know how much I was excited about that region, and you can see that there's increase in the high frequency broadband activity. This is the onset. This is the yellow, meaning that compared to baseline, there's some increased activity across all the different trials from those many electrodes across different subjects. But because we keep the subject as a variable, we can then ask something about the timing of those effects and how those might predict all kinds of things. In this case, we look at the timing of those effects across individuals. We were able to show that among all the participants, the timing of the latency was related to how well they did. That wasn't the case in kids. So the faster, the earlier latency among all the participants showed, distinguished them, allowed them to perform better, but that wasn't the case in those who had shorter latency also able to then go and ask questions about the medial temporal lobe, and kind of, for the interest of time, go fairly quickly just to show that when we look at, now this is kind of look at, of course, all frequency ranges. For what predicts subsequent memory, we found two different hotspots within the theta range. The theta is an important for memory. We had this fast and slow theta, and following up on that study, we were able to show that indeed we can quantify fast and slow theta in the brain of all our participants. We can also find that the fast get faster with age, and the slow get slower with age, among all the lectures we had from the medial temporal lobe. So we have something fundamental about maybe segregation of the theta range within this age range. That by itself didn't predict memory, but we went on and asked, do we find this similar pattern outside of the medial temporal lobe? And yes, everywhere we looked for, we found the same. So it might be some basic mechanism of maturation of the brain that kind of get those two theta rhythms to separate. But what's more important is that this whole study was to test something about how those regions coordinate. And we found that those two different theta rhythms had different roles in communicating or coordinating between the medial temporal lobe and the prefrontal cortex. And this fast and the slow differed such that the slow was related to the coupling between the power of those, and the fast was related to something that was about the synchrony between those two elements. Both showed specific effects that related to age earlier on in the trial. So now we know that it's probably something about getting ready to, or like being able to quickly do it. And again, this is just scratching the surface, but we were so excited to ask, do you have any more data from this subject? And sure enough, half of them had DTI. So we're able to link both of those age-dependent differences in the two different types of theta to go through the same white matter track, suggesting now we have multiple things, but we also can link them to some structural region in the brain. Okay, so with that, I was just going to say that there's kind of left you with kind of more of like excitement rather than the actual details, which is perfectly fine. It's kind of like roughly where we are still in that field. The data is exceedingly rare, and the type of insights we can get from this data are really enormous. So this is exciting. For example, we can learn more about the timing of localized neuronal activity that would allow us to differentiate stimuli from response-related effects. What I'm saying here now is that we can look at what is locked to the response, to the onset of the stimuli, or to the response, and we can find different things. Now we have some much better handles of how the brain organizes itself to respond. We don't just have like, okay, there was something on, and then they responded. We can actually lock it to that. We have been identifying local phase amplitude coupling and interregional mechanisms of coordination via rhythmic brain dynamics. So we kind of, the whole phenomena that we can quantify from brain rhythms or brain oscillations doesn't translate very well with fMRI data. So we have a whole new window on how the brain actually works. And then, as I said, there are multiple signatures that happen at the same time. And right now, one of the questions we're very interested in is try to understand this fundamental aspect. How does the brain develop through the neurodevelopment of different brain rhythms? And for that, we're actually partnering with people that do computational work and try to kind of take neurons, simulated neurons, and get the same type of effects that we find that might be separation of the theta rhythms and such. So very exciting. How is cognitive development linked to brain development? We can look at brain structure and link it to aspects of age differences in memory. With multiple measures of brain function and link them to age differences in memory. And moving forward, there's a lot of open questions, but surely what would help us a lot is stick kind of to theoretically guided mapping of multidimensional cognitive constructs, try to put a better handle of what is it in memory or cognition in general that develops, maybe go back into what is memory development, the development of, and how is different capacity, user strategy linked into all the effects that we find in the brain. And then try to close some gaps across levels of organization, such as how we converge across methods, continue to work towards harmonization across labs so we actually can aggregate data and get fundamental understanding that's beyond what each lab can have. I got this and I got that. And ultimately, and this is something that here in here in Dallas, we are very much minded for and working towards, is work towards increasing our samples, so we have more robust effects and get more dense sampling and have the same, and more than one person do things together. I would characterize it as a dense sampling. So with that, we hope that ultimately would be get better basic theoretical framework memory with some implication or clinical relevance, maybe some educational relevance, some broader societal effects, especially if we start to better understand the sources of individual differences that are both biological, genetic, and environmental. But ultimately, what vibes well with the work in here is understanding the modifiers of typical development across the life to promote health and well-being. And I will end with this, and then I'll switch to the other presentation. Thanking for many people, every other work they've done, of course, has been conducted with me by with many other people. What you see here, the current budding people that are having the lab here at UT Dallas, and some of the people I've been working and still are working with in Detroit, and many collaborators. With that, I'm done, but I know I switched this slide, so I need to go back to the original one. Just give me a second. Now I'm done.
Thank you. Thanks, Noah, for a very provocative talk. We have a few minutes for questions for Dr. Ofen.
Thank you so much for the presentation. It was wonderful. In the commentary that you discussed, one of the things that you mentioned was the environmental issue. And I wonder whether you are looking at any other determinants of healthcare that would affect the growth, the change in the brain.
So in my lab, we haven't focused on any specific population, but the project that I have been involved, not quite environmental, but to some extent maybe, even if you just think of preterm birth. So this is some project that we have a sample of preterm born individuals compared to children compared to term-born individuals. But what we have been doing in expanding what we do is getting through either questionnaires, some assessment of the socioeconomic status that parents report to, or what is getting better and better at taking census data from the zip code to characterize large samples or the samples that we have based on some different characteristics of it. So socioeconomic status can be within the household, but then we can look at the neighborhood index. And that's part of a stated goal we have to try to see whether the household, for example, is more important earlier on. Neighborhood becomes more important in adolescence. Maybe this, but once you have less of it. So we will be able to test exactly this, because now we have combined the data from across labs across the country that has allowed us to characterize both.
No, a really elegant presentation, and so fine-tuned. One of my questions is related to the EEG and the testing of the physiology. Do you feel like the brains of someone that's undergoing epilepsy is a good signal for the normally developing brain, and how do you kind of take that into account of this developmental part of?
Excellent question that I'm really well prepared to answer because it comes up almost every time by everybody, right? So the reviews of the grant, that was we had to very come up with a lot of very well-reasoned patterns. So a couple of things there. Those of you who would know people that live with epilepsy, you know that there's a really wide range, and if you know them, they could be perfectly normal, even like above normal. Like there would be a lot of people that, when it's well managed and not very severe, then there is a lot of debilitating when you have a seizure, but otherwise the child might have a very typical developmental trajectory. Perhaps even, my personal experience, even better, because there's so much care about trying to optimize a lot of things. But a couple of things. So first of all, to eligibility to participate in the study, we won't be able to test individuals that are very low performing. So we have only high performing individuals already, and that's a self-select. And we know they're high performing because they have some score of IQ and able to follow the
Has some sort of IQ.
IQ test, yeah. So
Okay,
we can say like within the normal
Because that
range.
was my other question, is how does this memory relate to kind of their overall intellectual function or academic performance, or have you looked at things related to more, how they're doing in real life?
So this is aim 3 for the grant. We haven't gotten this because we're waiting for the fuller sample. But because we do have some neuropsychiatric evaluations on those individuals, we can look for the magnitude or something with the measures we quantify from the intracranial data and try to see how those link to standardized measures. Right now we don't have specific plans for achievement to the extent that it's in some standard battery that we have that, but otherwise we haven't been following this. But so a couple of things. First of all, we take the high achievement. We are also we have some information about them, so the physician think about them as typically developing. But what's really important to also to kind of communicate and understand for these clinical practices, the one I identify with the seizure is, but in like for the minutes, so they can. But to find this, you also need to sample the normal tissue that is not seizure inducing, right? So the way that the electrodes are being placed are really not just to capture what the seizure is. You have to capture normal tissue. So what we do is when we get those data, we very, very painstakingly and carefully, and some people in my lab here have seen that around, very carefully removing or taking away from our analysis any electrodes that have abnormal activity in them. So we're staying only with the electrodes that we deem normal, as in there's no spiking, the physician didn't tell us it's abnormal. And even if there was some abnormal activity within a trial, then that trial is getting cut. So we left with really good data of typical brain behavior. And people have been using it for, I don't know, well, all the work has been done forever, right? But within cognitive neuroscience, it's kind of fairly more recent. Developmental, it's really nothing. Like it's really what we're doing is kind of bringing a lot of those to kind of specifically bringing it to basic cognitive neuroscience questions.
I think we're out of time, unfortunately. But I will speak to one last question. What was the IQ inclusion criteria for that?
So 80, but sometimes they can take 70. So, but there are really, really. So I have just kind of get. Remember here I said those blue dots are the intracranial patients. So you can see that they perform well, and the trend is the same trend as the individual. So this is the okay. And you still don't believe us? So look at that. now you think we're fine.
Thank you so much, Nora. So thanks for coming today, Lifelong Brain Health. This is our shared mission. Thank you again. We learned so much from you, and really amazed by that and inspired by the research you're doing. Thank you all for joining us this afternoon. We look forward to seeing you next week, where I, along with Tracy Brown from my lab, will be talking to you about cannabis, sleep, and memory. take the Great Brain Gain text challenge. There should be a slide with a QR code. And the next screen. There it is. Thanks, everyone. Have a good week.